like to introduce you to our first plenary speaker, Ravi Bellamkonda, who is a dear colleague and also a mentor of mine. Ravi Bellamkonda is a world-renowned scientist and expert with interests that intersect between biomaterials, nanotechnology, and their implications in neural repair, both in the peripheral and central nervous system, as well as tumor therapy and neural interfacing. Dr. Bellamkonda is a trained bioengineer and neuroscientist. He holds an undergraduate degree in biomedical engineering. His PhD training is from Brown University, as well as postdoctoral training at MIT. August 1st, he joined the prestigious School of Engineering at Duke University as the dean. But prior to that, he was a huge instrumental part of the biomedical engineering department, actually, here at Emory and Georgia Institute of Technology. He was the Wallace Coulter Professor and Chair of the department. And certainly with that, he's made some amazing contributions to the collaboration between Emory University School of Medicine and the Georgia Institute of Technology. Dr. Bellamkonda is also the president of the American Institute of Biological and Medical Engineering. He's the recipient of numerous honors and awards, which include the Clemson Award for Applied Research from the Society of Biomaterials, the Eureka Award from the National Cancer Institute, a Career Award from the NSF, and the Best Professor Award from the Georgia Tech BME students. So without further ado, please welcome Dr. Bellamkonda.
Thank you so much. Good morning, everybody. Thank you so much, Randy, for that introduction, and thank you also for inviting me. So I hope to tell you a little bit of a story today about sort of the evolution of my lab and the way we've approached peripheral nerve repair. And it fits very well with what we heard in terms of regenerative medicine and how regenerative medicine is evolving, as well as the convergence with rehab and the use of physical cues and mechanical cues to affect repair. Just want to start off by saying most of the work I'll talk about today was done by a very talented research professor in my lab, Nasser Mukarram, and also in collaboration with Art English, who's at Emory in cell biology. So the question is, it's an intriguing title in the sense of immune engineering nerve repair, and that's the evolution. When we started working on peripheral nerve repair, we started working on nerves. But it starts in the 90s when we thought of tissue engineering and regenerative medicine. The general idea was that it was, I don't know how many of you are familiar, many of you are quite young in the audience, but tissue engineering is a weird field in the sense that it was initially seeded not by NIH, not by basic scientists as most fields are, but it was seeded by industry. So in the 90s and late 80s, there was a flush of investment with the promise that we would grow organs in a dish and they'd come in different sizes and we'd transplant them, right? So it hasn't quite panned out this way. And in fact, I'd argue that the lessons from that whole story is that A, science is important, B, that we might want to pay more attention to endogenous repair rather than think that we can recreate complex environments in vitro and then sort of transplant them. So in a way, my lab's evolution charts this path. And hence, I'll talk to you about triggering endogenous repair being our focus. Now, many of you know peripheral nerve repair is caused by various things, and the picture that's not here is also a major contributor to creating peripheral nerve gaps is the resection of tumors. So when you have a tumor associated with the peripheral nerve, often when you resect it, you have to sacrifice it and you create a gap. And the gap and the healing of the peripheral nerve gaps, and I'll largely focus today on PNS, is actually an unmet need. Only 10% of folks that actually get peripheral nerve gaps actually fully heal in any way. It's a common misperception that peripheral nerves somehow heal themselves, and they do to a greater extent than central nerves, like spinal cord and such, but still the functional recovery is not fantastic. And not to mention that because it's slow, the nature of the target muscle changes, cortical control of the muscle changes, so it's complicated to get full recovery. You might also think, well, the reason we haven't made as much progress as we'd like is because it's a small market. And, well, small or large is relative. Just some numbers here. And it's not so small, actually. Extremity repair is a big deal. A lot of times we do sleeves and things as protection, if this works. So this is sort of a protection. You can just put a sleeve on. But this stuff is transected gaps. That's not too small a market. And there are not that many players in this market, actually. And so there is still opportunity here. So what are our options when you have a nerve gap? So if the gap is small, you can obviously try to coapt the nerves and bring them together and suture them. But you want to avoid tension, right? And that would injure the nerve. So often, depending on the nature of the injury, this is not an option. Although there are some studies you may be familiar with from UPenn and others where if you slowly, ever so slowly increase tension, you could stimulate growth actually. But that's practically not possible in a clinical setting. And so usually you can't do this. So the gold standard really for clinical repair is the use of an autograft, where you take your sural nerve from the back of your leg and use it as a bridge to repair your peripheral nerves. And there are issues with it, and the recovery is not perfect. You need a second surgery. You have loss of function where you get the nerve from, and such. And so the other approach, which many labs have been investigating for a long time, including ours, has been the use of nerve conduits. And the idea is, well, if we need a bridge, why don't we put a tube in there, put the two nerves in there, and put stuff in the middle that helps the nerve grow back instead of using the autograft as the bridge. And so that's been the approach. So unfortunately, to this day, we don't have a technology to design this kind of approach that matches the performance of autografts yet. And in a sense, it's not a fair comparison because autografts are not just the physical bridges, but you usually transplant Schwann cells that are in the nerves along with it. So this is a sort of combination therapy, if you will, whereas these things have largely been sort of by individual proteins and trophic factors and conduits, not really approaching the might of this yet. But we may be beginning to change that, and that's the story that I'll tell you today. So as an engineer, my thinking was, well, what do the autografts do, and why can't we just engineer the features that autografts have into my scaffold? And it's a little bit insulting that after 20 years, I still haven't been able to do this as well as we'd like. But I'll tell you how we're getting close. So how might we design a bridge if we wanted to bridge a peripheral nerve gap? Well, so theoretically, you could say, well, I need something to go in between. I have a proximal nerve end and a distal nerve end. And then you want to put something here, because nerves are anchorage dependent. They don't float, so they like to grow on things. So you can't just have a gap. And in fact, when you have a long gap, so we thought maybe we could create a scaffold. The scaffolds were mentioned earlier this morning in the workshop. So the idea is you have some kind of physical scaffold, a gel perhaps, that has the features to support three-dimensional nerve growth. And then you would introduce into the scaffold haptotactic cues or extracellular matrix proteins that when nerves see, they're stimulated to grow. Or you might introduce within it trophic factors like nerve growth factor or BDNF or some of the neurotrophin family proteins that might attract the nerve to come in or stimulate them to grow, and you'd get the nerve to bridge. Now, the other thing, for those of you who are not nerve types, although many of you I suspect are, the difference, and this afternoon you'll hear about bone repair. So in bone and non-healing bones, typically you have the callus formation from the periosteal layer, and the periosteal cells come in, and you're interested in a bridge, and you're mechanically trying to bridge the two ends. In the nerves, essentially you want the nerves to grow back, but then everything distal here degenerates because the cell bodies that support the nerves in the distal segment are up here. So in the nerves to get functional recovery, not only do you need the gap to be bridged, but you also need it to enter the right lumens on this side, go all the way, innervate the distal target, typically a muscle, central control of that because they get scrambled into which hole they go into and such. And so functional recovery is much more involved, complicated, takes longer than thinking of just bridging this gap. Okay, just wanted you to sensitize you to that. The other thing I'll tell you is that people often, again, if you're not a nerve type, between PNS and the CNS. So in the peripheral nerve, I believe that the challenge we have today in the field is the bridging of longer nerve gaps. So in a rat that's about 13, 14 millimeters or longer, up to 10 millimeters, we might sometimes get spontaneous repair. But in the CNS, if you have a gap that's even less than a millimeter or a millimeter, we have issues. So I think of this problem as bridges and dams. In the CNS, my belief is that the chondroitin sulfate proteoglycans that the oligos secrete form glial scar, and the challenge in the CNS is really overcoming scar tissue and the astroglial scar that forms primarily composed of chondroitin sulfates, and how we overcome that. In the PNS, I believe the problem is different. Although there are CSPGs in the autografts, and we could do better, largely the problem is of designing better bridges to bridge these long gaps. So just to delineate the challenges. So my lab then spent many years in the late 90s, early 2000s, designing these scaffolds to bridge nerve gaps. In fact, we got quite fancy. First, we spent many years asking the question, if I wanted to design a gel that supported nerves growth in it, what would the rules of that gel be? Do we know what porosity, what influence charge plays, what mechanical stiffness do these gels, should they be? And so it turns out that we didn't know many of these things. We knew that in matrigels sometimes you'd get nerves to grow. We knew anecdotally in collagen gels we could get nerves to grow, but we didn't know why or what properties. So we wrote a series of papers in the literature sort of investigating the rules for 3D nerve growth in terms of porosity, stiffness, charge, and such. And we settled on a gel at the end that is this picture. This is a dorsal root ganglia in culture, in vitro, sending out processes in 3D in an agarose gel. And agarose is just a carbohydrate gel. And it is very convenient to use for many reasons. It's a thermoreversible gel. So it can be a liquid, you cool it, it gels. Once it gels, it remains a gel. So it was very easy to handle. And we could ask questions about what proteins were relevant without the confounding issues of matrigel having God knows what in it, or in collagen gels, which already had proteins in it. So the idea was you wanted a blank template of a gel that structurally would support nerve growth, and then you would introduce as needed trophic factors or extracellular matrix molecules into this. Not only could we design such gels, and this is a picture of this. In fact, if I flipped this gel over, it would look just like this. It truly supports three-dimensional growth in gels, and we spent quite a bit of time trying to design these gels. But we also got quite fancy. We made gradients of this from the proximal to the distal end. The idea is to bridge gaps. You could make gradients of various kinds. So this is an example of a trophic factor gradient that we made. Essentially, you take a protein of interest, you make a photoreactive component to it, and you let a diffusion gradient develop across the gel, and then you shine light to freeze that gradient in place, to couple it to the backbone of the gel. So we would play tricks like this to make gels that had gradients to entice nerves to grow back across long gaps. So what happened with this whole study was that across 10 millimeter gaps, we showed that a combination of these extracellular matrix molecule gradients plus trophic factors, usually NGF or BDNF, would actually come very, very close to meeting the performance of autografts. So we were very excited. We went on to extend the gap to longer gaps, to 15 millimeters or so. So when we did that, something actually quite disappointing happened. So after 10 years of work optimizing all these things, when we went to a 15 millimeter gap, we found that only 2 out of the 10 animals or so that we implanted with such optimized materials actually resulted in successful bridging of the gaps. Okay? And this was quite disheartening in many ways, because we had spent 10 years doing this. So, I joke that, like any good scientist, I wrote another grant. So, and that's what that's what we did this is the next 8 years of the journey, if you will. So one of the things we did was that we noticed when making such gels that we happened, don't ask me why, I happened to put a 10O monofilament nylon suture into one of our gels that we had spent all this time optimizing. When we did, in fact, this is a picture of that gel fiber. This, you open the suture 10O and you put it in. We noticed that even in these hydrogels that we had spent so many years optimizing, if you give a DRG or a PC12 cell, whatever cell you work with, a choice, they tended to grow on this fiber, and then when the fiber ended, they happily hopped off and went into this gel that we kind of spent so many years optimizing. So then we started thinking, maybe we shouldn't fight this. So it turns out that if you look at our cortical development, right, so we have 6 layers, and they're born, the cells are born in this ventricular zone, and then you have this radial glia that are set up developmentally. And it turns out Connie Sepko and others have done beautiful work on looking at the birthday of the cells that make up our cortex. And it turns out they're born in the ventricular zone, and then they migrate along these fibers and hop off as the time varies. So the older cells hop off soon. The younger cells that are born later have longer distance to go before they hop off. And this is how our cortical organization is actually set up developmentally. So, and Pascal Rakic and others have called this the glial monorail, if you will, actually, this pathway. Not dissimilar from this. And this is another picture. So anyway, so we thought, okay, maybe we should try to incorporate some of these features into our bridge, and what would that look like? So we went to an old technique from 1928 called electrospinning. So you take a polymer and you create an electric, and you pick the right solvent for the polymer, a plastic, if you will, and this lends itself to many kinds of polymers. So, and you shoot it out of a nozzle, this solvent and the polymer in solution across a large electrical gradient. And then you can, there are movies on YouTube, you can do electrospinning, and you can see it shoots out a fiber, and you have a target that it collects on, and you can make films out of this electrospinning technique of fibrous nature. And by controlling the distance and the voltage and the solvent and the polymer concentration, you can make various diameters of fibers of actually multiple kinds of polymers. It turns out if you play a trick, and instead of a static target, you have a drum, and this is the home of Home Depot, so we actually literally went to Home Depot, built a drum. And so in the target, instead of a static thing, if you have a drum that's spinning, you actually have something like spooling, and you can make films like this with oriented fibers. All right? So we made this out of polycaprolactone, which is degradable, polysulfone, pan-PVC. So there's a bunch of polymers. I won't throw acronyms at you. But you can make such films. And then you can take one of these tubes and put the film in the middle with the axis of orientation going through the tube. And the idea is, if I give this option, would nerves march along and grow along these tubes? And then I could put the gel around it, and now I have really trophic factors and ECM molecules and physical cues. Now can I get it to repair 15-millimeter gaps? So as a control, because we had done all that work, we said, well, we first need to understand what does the fiber do by itself before we introduce all these other factors. And so we did a short, what we thought of as a pilot study. And for the students in the audience, I always tell my students, whenever you do a pilot study, don't do it with sufficient N so you can actually use it for a paper if something good happens. So, which we tend not to usually for a pilot study. But anyway, this student, Nasser, listened to me, and something remarkable happened actually. So we picked a 17-millimeter gap. This was actually Youngtae Kim's work, actually, I apologize. Youngtae Kim and Isaac Clemens. So across a 17-millimeter gap, just with this fiber, 10-micron film with oriented fibers, we got bridging of nerve gaps that we did not see even when we put our 10-year engineered hydrogels in the gap, which is, again, kind of, I don't know. So we thought about this. So it's like, what is going on? Because this is the polymer wall of the polymer. You're looking at a longitudinal section. This is NF160 staining from the proximal end to the distal end, complete bridging of a 17-millimeter gap. In fact, the first instance that we observed, Schwann cells come in. When you have a randomly oriented fiber, it doesn't happen. It only happens when you have oriented fibers. All right? So this got us thinking a lot about, what was going on. And Isaac went on to do his entire thesis on this. And what he found out was what's happening in this case when you have oriented fibers is that you have efficient migration of Schwann cells into the gap from both ends. And the Schwann cells then make the trophic factors and the ECM molecules and all this stuff that you need, and in a sense, help the axons follow, and you get regeneration to happen. So this was a sort of aha moment in the lab where we had spent 10 years thinking about nerve repair and hence building bridges and materials optimized for nerve growth. And suddenly, by happenstance, we happened upon an approach which did not actually work via nerves, in a sense. It worked via Schwann cell migration into the gap, which then had the nerves to do things, right? So we had never, actually, in my gels, which I admit, which was kind of stupid in looking back, never actually cultured Schwann cells in our gels, had optimized them in any way for Schwann cell migration, differentiation, production of trophic factors. We had done none of that. Why? Because we were very focused on nerve repair, and we optimized all our things for nerve growth and nerve repair. And here was an instance, by luck and happenstance, where we noticed that Schwann cells actually, if that was the target, we would have these positive effects. And it really was an aha moment. We got a little bit of a write-up in Science for this. So because it was the first instance of without exogenous factors, like the trophic factors I alluded to, you would bridge a long gap. So then Sir came into the lab, and I see Art has joined us. Hi, Art. And he asked the question, well, if Schwann cells are the key to this, could there be other cells that we could target, including Schwann cells, that might then help us out and bridge these gaps? And so that would be a logical extension of the finding that we had initially, that Schwann cells were an interesting target. And so the idea was, initially, you tried to recreate the complex embryonic environment of trophic factors and gels and such, and so you're going higher up now, where you say, instead of that, we had found that maybe we could just release NT3, which would attract Schwann cells, or a few topographical cues, and you'd get this. But if I progressed logically from here, is there some other master regulator I could target that would orchestrate all of this and get endogenous repair? So if you look elsewhere, so suddenly we start paying attention to when does repair happen, how does, in species where you do have fantastic repair, like the salamander, how does this work and such. And so that led us to the whole immune story, and I'll tell you. So this is an interesting paper. I had the reference here. It's disappeared, maybe down below. So where you know that they regrow their entire limb. You've seen videos of this, I'm sure. So when you cut off the limb, the entire thing regrows. And it regrows by the formation of this thing called blastema at the end, right? So what this paper did, and we've known that for a while, what this paper did was, there's a standard way of depleting macrophages by making liposomes with a drug called clodronate, and you give this thing and selectively kills all the macrophages. What they showed was that if you resect the limb and treat the animal with these clodronate liposomes, there's no blastema, no repair happens. If, however, you do the cut and delay and deplete macrophages later, post-blastema formation, you still have the limb and budding and the formation happen. Again, a very clear instance of the role of macrophages, which we generally think of debris cleaning, scar producing, inflammatory cells, right? That's our general, at least that was my view of macrophages, that the role that they play in the repair of this in a salamander. Indeed, when we further looked, there is literature now, rich literature, talking to you about macrophages not being all the same. There are many kinds of macrophages. In fact, there's a whole spectrum that are A, B, M, they're called M2, or the so-called alternately activated. So there are two major categories. One is this classical macrophages, which I alluded to earlier, and the other so-called alternately activated macrophages, so they're called M2 phenotype. And Steve Badalak and others at Pittsburgh also cells for a while. So where there are subtypes going up to E now. There's M2D and E and others. And the idea is that these are sort of reparative macrophages. Some are regulatory, the M2Bs, but these are reparative macrophages, and there are these M1 macrophages. And they're not all the same. And in fact, if you look closely, in wound healing and such, initially after injury, if you look at the genes that are expressed by these macrophages, largely they are inflammatory, so the so-called M1 macrophages. But over time, when you look at healing of nerves, or of wounds, it turns out that the switch in the gene expression from largely M1 and a little bit of M2 to the late stage of tissue remodeling, where very few M1 genes are expressed and mostly M2 genes are expressed. In fact, it is thought now by some, including our lab, that this switch from the initial inflammatory response that you do need to trigger healing of any tissue, not just nerve tissue, and indeed this study is from wound healing, is necessary for you to get a resolved injury and get healing instead of scar. If you remained in the M1 state for whatever reason, you would get scar. If you were able to switch the macrophage phenotypes to the M2, then you get healing. So that this switch is important. And in fact, there's some thinking that in diabetes, when we have impaired healing, that it is the switch that's actually goes awry, that the switch is somehow doesn't happen in diabetes. Okay? So the thinking is this, that you have an inflammatory event, and what you need to do in some ways to get healing is to make sure that a sort of balance between inflammation and the timing of inflammation to healing types of macrophage modulation may be important in general for tissues. We didn't know if this applied to nerves per se, and we started looking at literature, and actually more recently we found this study that indeed even in nerves, in peripheral nerves, if you look at the population of cells over time, and this is a study that was published in 2011, that indeed initially you have neutrophils and M1s, and then later you have M2 macrophages. Now this could be macrophages coming in later, or some of these macrophages being converted to become M2 macrophages, right? So this is a differentiation of the same macrophages taking this phenotype. Indeed, I'll show you that that's possible. So either these cells are different and coming in later, or it is some of these cells switching from, after cleaning up their debris, to becoming more healing type of macrophages, okay? So Nasser went on to ask the question, well, could we play with this system? Just as we had with our oriented fibers, optimized for Schwann, we do something to modulate this system, and would that result in nerve repair across long gaps? And that's the data I'll show you next. So it turns out, experimentally, there is a way to bias macrophages that you isolate either to the M1 phenotype or the M2 phenotype. And one way to do that is if I expose these macrophages to IF and gamma, I typically get M1 macrophages, and if I expose them to IL-4, I get typically M2 macrophages. So the link between M1 and M2 macrophages and actually how nerve repair happened, peripheral nerve repair happened, wasn't that clear to us at this point. So what Nasser did was, well, is there a possibility that they affect Schwann cells in some way? Because we had found Schwann cells to be so critical for nerve repair in our earlier study with Isaac. So what he did was he took conditioned media now of taking the same cells, biasing them one way or the other, taking conditioned media from these cells, the M2 cells, or conditioned media from these cells, the M1 cells, and putting them on Schwann cells and studying, does their migratory profile change? And lo and behold, indeed, if you have IL-4, which is this conditioning that results in the M2 macrophages, something that they produce actually causes a vast increase, a significant increase in Schwann cell migration, right? Compared to IF and gamma, or just treated in tissue culture medium. However, if I take IL-4 or IF and gamma, directly add it to Schwann cells, right? Not to macrophages, there's no effect on Schwann cells, right? So this is an effect mediated by macrophages because the directly IL-4 or IF and gamma don't do anything to change Schwann cell migration, right? So mediated through these cells. Okay, so we said, okay, let's try in vivo, and how does this work? So what Nasser did then was he took one of our gels with no oriented fibers or anything, no extracellular matrix molecules, but he just mixed in. We have all sorts of things in the lab for controlled release over many weeks. We have all sorts of fancy drug delivery techniques. But in this case, the whole idea was you're trying to trigger an endogenous repair pathway. And by going to try to modulate macrophages, the whole idea is soon after injury, if I somehow bias the macrophages towards the M2 phenotype, would I get effects that are longer term? So you don't want chronic release of proteins in this case because your hypothesis is that you're trying to tap into endogenous mechanisms and you just want to bias it one way or the other in that fulcrum spectrum and have the pathways take care of themselves. So this way you wouldn't have to guess whether L1 is more important or laminin is more important or NGF is more important or NT3 or NT5. We don't have to fight these molecular wars, if you will, right? So that was the idea. So the idea was you just mix it in the gel, and typically when you mix it into the gel, within 6 to sort of 12 hours, it's gone. The protein is released because there's nothing really keeping it there, right? Within 6 hours, it's gone. And it serves our purpose, so we explicitly did not do any prolonged release into this. I'll come back to the Fractalkine story in a little bit. So the idea was you release either IL-4 or IF and gamma in the gap, bias them one way or the other, and see what happens to repair across in this case, I think it was a 15 millimeter gap, and after 3 weeks. And indeed, we did get repair when we when we looked at IL-4 all the way, did not get repair in the control or the IFN gamma case. And this shows you S100, which was a Schwann cell stain. In the IL-4, we had more Schwann cells in the nerve gap. We did it a little bit closer to the distal end at 3 weeks. And if you looked at axons, indeed, there was no comparison. We had mostly axons at this location of the gap, this being proximal, that being distal, only in the IL-4 case. Stark, okay? Now, this was gel, something released only for the first 6 hours after injury. No other fancy engineering. And indeed, if you looked at the cells inside, you can stain for the cells in the gap in the 3 conditions and look for activated macrophages, the M1 macrophages, and the regenerative macrophages. And what we found was that essentially the best correlation to this effect that we saw with axons at 3 weeks in the nerve gap was not with the total number of macrophages, which were kind of similar. I'm sorry, the bottom thing is cut off. Or the M1 macrophages or M2, but the ratio of the M1 to the ratio of reparative macrophages to the inflammatory macrophages. So the thing that correlated best with this outcome of axons growing was not the total number of macrophages. Had I just stained for macrophages, I would have seen no difference in either case. But if I look closer into what kinds of macrophages are there and what's the balance between the M1 inflammatory macrophages and the reparative macrophages, that balance is the best predictor of whether I saw axons in the gap or not. Then we got thinking, is this only happening when we do IL-4 and IFN gamma, or is this generally true? It turns out in the lab we've done many things over the years looking at nerve repair at exactly the same time point, and we looked more closely. It turns out we've done controls, IL-4, IFN gamma. We had just done the films. We had done autographs. It turns out that independent of what we did or didn't do in the gap, this ratio of reparative macrophages to inflammatory macrophages is a high predictor of whether you have axons in the gap or not, independent of what you do. Okay? So this is not just when you artificially manipulate with the delivery of IL-4 and IFN gamma. Independent of what we did, and this is a set at the same time point, there is a correlation strong between the number of axons you see growing back and this ratio of reparative macrophages and inflammatory, and towards the direction that as you tip this balance towards the numerator, in this case, which is the reparative macrophages, you get more axons growing. Right? So this is pretty powerful. So NASA then, being relentless on that thing, asked, where do these cells come from in macrophages? It turns out, because he was delivering IL-4, and I wanted him to do a chronic study of long-term looking at functional recovery and all of these things, because I told you that's important. But before, he said, okay, is IL-4 the best way to do this? And we started asking questions. It turns out this picture is a little bit complicated, but still emerging. So it turns out monocytes are the first cells that are recruited to injury site. There are some tissue-resident macrophages, but mostly they're monocytes recruited from the blood that are recruited here. And then these monocytes can differentiate into the inflammatory monocytes or the sort of reparative monocytes, which then differentiate into inflammatory macrophages or reparative macrophages. So the circulating leukocytes actually come before the macrophages. There are some, as I alluded to in many tissues, so-called tissue-resident macrophages, but there are not that many of those, okay? Mostly when you have inflammation and injury, they're recruited from blood, these cells. It turns out that there is a factor, Fractalkine, that biases the recruitment of monocytes to be in the reparative phenotype that then biases it towards this. Now remember, my whole story is that of endogenous repair. And endogenous repair, I'm telling you, the take-home message is there are cascades like this that are multiplied, biased initially what happens after injury. And the idea, and you can see one such cascade. Now, these things then make maybe laminin and NGF and BDNF and God knows what else to help us, and maybe basic FGF to have blood vessels grow. So they make all sorts of things. So the idea is to do this bias. So then he asked the question, should we be looking at Fractalkine instead of IL-4, and how do they work relative to each other? And it turns out, actually, this monocyte bias thing is also true in cardiac tissue. People have shown that initially you have these monocytes after cardiac injury, inflammatory monocytes, which later switch over to these sort of reparative monocytes in cardiac tissue when you do get healing after some injury to cardiac tissue. So there was literature suggesting that this story actually might work, but again, there was no information in the peripheral nerve case, and so we are. So the idea is this. So you have a conduit now, and you want to release something here to bias things towards. Nasser likes drawing pictures, so I have to show you this. So there's reparative macrophages, inflammatory macrophages, fractalkines, and the sort of markers of these cells. And the goal is to release either IL-4, which is the best case scenario we had seen earlier in terms of macrophage phenotypes, or this new chemokine, fractalkine, which biases monocytes towards the reparative monocytes, which then talks to macrophages. And so here we are. The idea is to recruit these cells and have these cells either preferentially become macrophages or monocytes that are reparative, or to differentiate them in that direction. So this is the study now, 4-week study with fractalkine, comparing it to IL-4. That's those two things here. Same kind of scheme, 15 millimeters, where you get no nerve repair in a rat if you don't do something in the middle. So there's no spontaneous growth. So it's a critically sized nerve gap. Okay, so what happens then? So we did many, many kinds of things. Bear with me. Still I'm doing okay with time, I'm hoping. So this is looking at the so-called reparative monocytes. Indeed, you get more with fractalkine than IL-4. This is regulatory macrophages. You get more with fractalkine than IL-4. IL-4 was our gold standard because we're trying to figure out what's the best way to do this for our chronic study. You can look at just total number of macrophages. Again, this is what you would have done maybe 10 years ago. You'd actually see that there are more with IL-4 than fractalkine. And if you look at healing macrophages, you don't see that much difference. But if you take the ratio, you see that the ratio, which I alluded to in the regenerative bias argument earlier with my graph, if I look at the ratio, indeed, IL-4 does okay, but the fractalkine treatment does way better in terms of the healing cells versus the sort of scarring cells, if you will. This then tells you about Schwann cells in the gap, which I told you are important, so more Schwann cells in fractalkine. And this is RECA stands for new blood vessels and endothelial cells that are in new blood vessels. And indeed, you have more in the fractalkine treated cells. If you look at the actual nerves growing back, and you have this is 4 weeks again, much better cable, larger, with number of axons also approaching about 2,000 or so. If you look at what happens in an autograft case, it's actually autograft gets a little bit more. It turns out that in general in these nerves, you have about 2,000 to 2,200 axons in the control. So this is just to give you a scale of how many nerves are growing back. Okay, so you have about 2,000 to 2,000, and it's not uncommon to actually have more grow back, and then they'll be pruned when they don't make the connections eventually, right? So it's not uncommon when you look in the nerve gap to see more fibers than the original nerve had, because there's branching and there's extra things. It's a strategy that nerves typically use. You send out more, you make the right connections, you get retrograde support, and you prune the rest. So that's not okay. That's fine. The other thing we wanted to check here is it working the way we thought? And I told you, remember I told you the salamander story with knocking out the macrophages. So we did the same thing with claudinoid liposomes and showed that if you look, and you have to do this early because this is at 10 days now. Sorry, I'm switching times back and forth. But indeed, if you knock out the macrophages, the effect that you observe with fractalkine goes away. So this is repair happening, and you get stunted repair when you knock the macrophages out, right? So it's working not by directly stimulating the nerve, not by blood vessels in and doing something you don't know. If you knock off the macrophages, it doesn't work. So then we said, okay, now we've got fractalkine. Let's see how fractalkine, which does better than IL-4, which does better than just the fibers, can we do fractalkine and see how it does against the autografts, which was our goal all along. So that's the story I'll tell you now. And this is where Art English comes in, because we did this study in his lab, literally. We couldn't have done it without him. So we have empty tubes, fractalkine tubes, or autografts. And for autografts, autografts, what we did was you would resect a gap, you'd flip the segment and suture it back, right? We didn't do sural nerve and such. So you take a sciatic nerve, you cut out a gap, and you flip it and you suture it back, so you have a nerve bridge. So it's not exactly. This is a set from Art's lab, and the idea is that you cut it, and then you put electrodes on this side and on here, and you see a way to look at repair. And here's some data. This is the number of axons. This is at 4 weeks. I thought we did a 14-week study. Well, I still have 4-week study, and I'll show you the 14-week study in a second. So at 4 weeks, you do, when compared to autographs, this is what you see. You do better with Fractalkine, but autograph does even better. This is a 4-week study. This is 14 weeks. I apologize for flipping things over. So this is at 14 weeks. If you look at the quality of axons with autograph, Fractalkine, and control, looking at axon density or myelin thickness, actually we do pretty well with the Fractalkine with this. And this is the electrophysiology with the M reflex, which is your direct stimulating proximal and looking at your muscle, or the H, which is through your sensory fibers and through a monosynaptic response. And it's not quite as good as autographs, but it's approaching that of autographs. And it's the best we've seen in our hands or in the literature in terms of bridging gaps larger than 15 millimeters. So my feeling is we're coming close, or we've come the closest with this approach than all the fancy engineering we did 10 years ago by trying to modulate monocyte recruitment. And we are nowhere near, I believe, optimizing for monocyte recruitment. There's timing issues. What happens if you have a chronic injury? So we don't know many questions. But the approach of all the ones we've tried in the lab and in the literature, I believe this approach has come closest to mimicking an autograft performance. Remember, this is just releasing something for 6 hours. At the time of surgery, you could inject something and you're done, as opposed to autografts or cell transplantation or stem cell therapies or Schwann cell implants or all these other things, right? So you're competing against an autograft which has cells, Schwann cells in it, which is pretty remarkable. So I'll just end and leave some time for questions on this idea that mimicking and biasing the early inflammatory response could be productive, and this balance, and maybe there are other ways. Maybe electrical stimulation, you'll hear from Art and others, and we haven't quite looked. Maybe electrical stimulation may bias the M1, M2 things. There may be other tools to increase your M2 to M1 ratio, and we're open to, we hope people will try those, that this is a productive way to go, and that this is mediated by your M2 macrophages through the regenerative bias argument that I showed you, and recruitment of reparative cells to the nerve gap early has dramatic effects long term. 14 weeks later, the electrophysiology that I showed you and some of the myelin quality data I showed you is 14 weeks later. So you can have long-term effects which are quite dramatic from a very early intervention because you're riding this cascade of the reparative cascade as opposed to the scarring cascade. So with that, I'll end and thank many, many people in the lab and funding and INDS just gave us a new grant for the immunoengineering approach and wonderful collaborators like Art without whom we couldn't do this work. So I'll pause there and take questions. Thank you.